Good day everyone, my name is Cristal Binyanes and today we will going to report our assigned topic, which is the development of digestive system from primitive mesenteron, the digestive tract. So digestive system, it provides nutrients for cell maintenance growth, consists of muscular tube called digestive tract or also called as gastrointestinal or GI tract or alimentary canal. So digestive system supports tissue with no direct contact with the environment. The GI tract is a series of hollow organs joined in long, twisting tube from mouth to anus. So the hollow organs that make up the GI tract are mouth, esophagus, stomach, small intestine, large intestine, and anus. The digestive system breaks down food into nutrients such as carbohydrates, fats, and proteins. They can be absorbed into the bloodstream so the body can use them for energy, growth, and repair. Unused materials are discarded as feces. So, formation of primitive gut tube. So, gastrulation is defined as an early developmental process in which an embryo transforms from one-dimensional layer or bastula and recognizes into multi-layered and multi-dimensional structure called gastrula. Gastrulation derives a three-tissue-layered organism composed of endoderm, mesoderm, and ectoderm. Each germ layer corresponds to the development of specific primitive systems during organogenesis. In addition to setting the embryo up for organ formation, gastrulation provides a mechanism to develop a multi-leveled body plan that demarcates anatomical axis formation with dorsal, ventral, and cranial and caudal axis. The cell and bastula rearrange themselves especially to form three layers of cell. The process is called gastrulation. During gastrulation, the bastula folds in on itself and cell migrates to form three layers of cell in a structure, the gastrula, with a hollow space that will become a digestive tract. Each layer of the cell is called germ layer and will differentiate into different organ system. So we have here an illustration. Around 16th day of human development, embryo begins to fold and the ventral surface become concave in two directions. The sides of the embryo folds in on each other and the head and the tail fold forward one another, forming a primitive gut. So the gut is the endoderm-derived structure. At approximately 16th day of human development, the embryo begins to fold ventrally in two directions. The sides of the embryo folds in on each other and the head and the tail folds forward one another. The result is that a piece of yolk sac, an endoderm-lined structure in contact with the ventral aspect of the embryo, begins to detach and become primitive gut. The yolk sac remains connected to the gut tube via vitelline duct. So around 18th day of human development, cranio, caudal, and lateral folding causes the opening of the gut tube to the yolk sac to draw close like persisting, forming a pocket towards head and the end of the embryo called the anterior or cranial intestinal portal, and a posterior or caudal intestinal portal towards the tail of the embryo. So around 22 days of human development, further folding and growth of the embryo causes the communication of the gut with the yolk sac to continue to get smaller, and the regions of the gut, foregut, midgut, and hindgut becomes further redefined. So by the end of the first month, the stomach bulge is visible. Dorsal pancreas has begun to bud. Connection of the midgut to the yolk sac is reduced to a yolk stalk. So connection of the midgut to the yolk sac is reduced to the yolk stalk, and the very thin vitelline duct. We have here lateral folding of the embryo completes the gut tube. Mesodermal layer of the gut tube is called planchnic. Visceral mesoderm derived from the lateral plate mesoderm, and somatic mesoderm lines the body cavity. So in letter A, the intraembryonic cavity bored by visceral and somatic layer of the lateral plate mesoderm is in open communication with the extraembryonic cavity. So in letter B, the intraembryonic cavity is losing its wide connection with the extraembryonic cavity. In letter C, at the end of the 4th week, visceral mesoderm layers are fused in the midline and form a double-layered membrane or dorsal mesentery between right and left halves of the body cavity. Ventral mesentery exists only in the regions of spectrum transversum. So mesenteries, portions of the gut tube and its derivative, are suspended from the dorsal and ventral body wall by mesenteries. Doubled layer of peritoneum that encloses an organ and connect to the body wall. Primitive dorsal and ventral mesentery. The liver is connected to the ventral abdominal wall and to the stomach by falciform ligament and lesser of respectively. The superior mesenteric of the artery runs through the mesentery proper and continues towards the yolk sac and the vitelline artery. This is gastrointestinal or GI tract system involves 3 germ layers, so mesoderm, endoderm, and ectoderm. So the mesoderm gives a rise to the connective tissue including the wall of the gut tube and the smooth muscles. So the mesoderm layer becomes circulatory system, lungs, epithelial layer, skeletal system, and muscular system. And then the second one is the endoderm. It is the source of the epithelial lining of the gastrointestinal tract, liver, gallbladder, and pancreas. So endoderm layer becomes digestive system, liver, pancreas, lungs, the inner layer. And the last one, we have the ectoderm. Further separates into the surface of ectoderm, neural tube, neural crest. The surface of the ectoderm is precursor to the epidermis, lens of the eye, nails, and hair. The neural tube differentiate into the brains and the spinal cord. The neural crest is in the source of the peripheral nervous system, including the neurons of the GI tract, also called as enteric nervous system. So the ectoderm layer becomes the hair, nails, skins, and nervous system. So the gastrointestinal system has division: the foregut, midgut, and the hindgut. The foregut or the anterior gut is formed the oral cavity to the initial part of the duodenum. The midgut is formed the mid-duodenum to the initial 2/3 of the transverse column. The hindgut is formed later 1/3 transverse column to the upper portion of the anus. So the 3 sections of GI tract have different blood supplies. The 3 sections of GI tract have different blood supplies. The foregut receives the vascular supplies by the celiac artery. The superior mesentery artery supplies the midgut, and the hindgut gets the supply from the inferior mesentery artery. So the foregut gives a rise to the esophagus, stomach, liver, gallbladder, and bile ducts, pancreas, and upper duodenum. So we have here the esophagus. During the 3rd week of the gestation, the respiratory diverticulum or the long bud forms as an outgrowth from the ventral wall of the proximal foregut. While the long bud continues to expand, it becomes separated from the foregut, which forms esophagus. Initially, the esophagus is short but becomes rapidly elongated as a result of growth and the relocation of the heart and the lungs. So when the embryo is approximately 4 weeks old, the respiratory diverticulum or the long bud appears at the ventral wall of the foregut at the border of the pharyngeal gut. The tracheoesophageal septum gradually partitions this diverticulum from the dorsal part of the foregut. In this manner, the foregut divides into a ventral portion, the respiratory primordium, and the dorsal portion of the esophagus. So the next one we have the stomach. During the fourth week of the gestation, the rudimentary stomach appears as a fusiform shaped dilation of the distal foregut. Subsequently, its appearance and position drastically changes. The latter can be better understood by visualizing a longitudinal axis of an anterior-posterior axis around which the stomach rotates. So the stomach rotates 90 degrees clockwise around its longitudinal axis, causing its left side to face anteriorly and its right side to face posteriorly. Hence, the left vagus nerve, initially innervating the left side of the stomach, now innervates the anterior wall. Similarly, the right nerve innervates the posterior wall. During this rotation, the original posterior wall of the stomach grows faster than anterior portion, forming the greater and the lesser curvature. So we have here a closer look at the mesenteries. So the stomach and the liver are suspended in a mesenteries that is attached to the dorsal and ventral body walls. So the dorsal mesenteries of the stomach becomes the greater omentum. The ventral mesentery of the stomach or the dorsal mesentery of the liver becomes the lesser omentum. The ventral mesentery of the liver becomes the falciform ligament. The rest of the GI tract is suspended in a dorsal mesentery, the mesoduodenum, mesocolon, and etc. So that's all for my report. Thank you for listening.
Hi everyone, this is Elame Iman Pacheco, the second reporter, and today I will be discussing the liver and biliary apparatus. This topic is the continuation topic of Miss Crystaldo Nunes, which is the first reporter. So, liver and biliary apparatus. The development of the liver begins during the fourth week of gestation, with the appearance of the hepatic diverticulum or the liver bud as an outgrowth from the ventral wall of distal foregut. The stalk connecting the diverticulum and the foregut narrows and form the bile duct, whereas the gallbladder and the cystic duct form as a ventral outgrowth from the bile duct. Initially, the bile duct opens anteriorly into the duodenum but ends up posteriorly due to the rational change of duodenum. So, the liver develops from an endodermal outgrowth, hepatic diverticulum, at the cranioventral portion of the foregut. Mesoderm surrounds the diverticulum, septum, and transversum. The hepatic cells or hepatoblast, both hematopoietic and endothelial precursor cells, then migrate into the septum transversum. The endothelial precursor cells or the Wittlein veins are surrounded by hepatic cells forming the hepatic sinusoids or by potential hepatoblast, give rise to both cholangiocytes and hepatocytes. The hepatoblast in mesenchyme closet to the portal vein form a bilayered structure. The ductal plate, these cells are remodeled to form bile duct in the intrahepatic portal tract, and the gallbladder and bile duct begin as cystic diverticulum. The gallbladder is initially solid and becomes cystic. Intrahepatic bile duct develops starts at the ileum and progresses to the periphery of the liver. The common bile duct form in an area narrowing between the foregut and the hepatic diverticulum. At birth, the most peripheral intrahepatic bile duct are immature with persistence of ductal plate. Maturity of intrahepatic biliary tree is achieved by 4 weeks of life. Now let's proceed to pancreas. The pancreas is formed by two separate pancreatic buds that later join. Specifically, the dorsal pancreatic bud forms as an outgrowth from the dorsal wall of duodenum, whereas the ventral pancreatic bud forms as an outgrowth from the ventral wall of the duodenum along with the bile duct. When the duodenum rotates and takes its C shape, it forms and pulls the ventral pancreatic bud dorsally and inferior to the ventral pancreatic bud. Subsequently, the pancreatic buds along with their rudimentary duct system fuse to give rise to the pancreas. Pancreas development begins during the 4th or 5th week of gestation as distinct dorsal and ventral buds arising from endoderm of the caudal foregut. The proximal duodenum, the dorsal bud, is larger than and is slightly more cranial to the ventral bud. Each bud communicates with the foregut through a duct. The rotation of duodenum causes the ventral pancreatic bud to rotate clockwise to the left of the duodenum and bring its posterior and inferior to the dorsal pancreatic bud. The two buds fuse to form the pancreas during the 7th week of gestation. The ventral bud forms the inferior part of the head of the pancreas and the uncinate process, and the dorsal bud form the superior part of the head. The body and tail of the pancreas, the ductal system of the two bud, fuse in the 8th week. The main pancreatic duct, which enters the duodenum at the major duodenal papilla, is formed by the longer dorsal duct draining into the proximal ventral duct to form. If the proximal portion of the dorsal duct remains, it forms an accessory duct that opens into a minor accessory papilla located about the 2 cm above the main duct. So the accessory duct opens into a minor papilla in 33% of people and ends blindly in 8% of people. 50% of people do not have an accessory duct. Endocrine cells are identifiable by 8th week. The exocrine pancreatic development continues after birth with maturation of specific digestive enzymes. A variation in this process may result in an annular pancreas which can constrict the duodenum. Also, since the dorsal and ventral pancreas arise by different mechanism, it's possible that one or the other may be absent in the adult. Next is the midgut. The midgut gives rise to the distal half of duodenum, the jejunum, the ileum, the cecum, the appendix, the ascending colon, the proximal two-thirds of the transverse colon. The lower duodenum, cranial portion of the midgut, the jejunum, ileum, cecum, appendix, ascending colon, and proximal two-thirds of transverse colon develop as the midgut loop herniates through the primitive umbilical ring during umbilical herniation at week 6. The loop then rotates 270 degrees counterclockwise around the superior mesenteric artery and returns to the abdominal cavity. So, the distal duodenum, jejunum, ileum, and cecum, appendix, ascending colon, and proximal two-thirds of transverse colon develop from the midgut between the 6 and 10 weeks. The midgut loop herniates through the primitive umbilical ring. So during the umbilical herniation at week 6, by 10 weeks of the development, abdomen has enlarged so that the entire length of the midgut can be accommodated. Following a 70-degree counterclockwise rotation around the superior mesenteric artery, the the bowel returns to the abdominal cavity, the large intestine returns following the small intestine, and thus an additional 180-degree counterclockwise rotation. Colonic fixation occurs after the return to the abdomen. Cecum and appendix begin as a diverticulum around the 6th week. The unequal cecum growth leaves appendix medial to the cecum. Next is hindgut. The hindgut gives rise to the distal third of the transverse colon, descending colon, sigmoid colon, rectum, the upper two-thirds of the anal canal, and partitioning of the cloaca. The cloaca is the endodermally lined cavity at the end of the gut tube. It has the verticulum into the body stalk called the allantosis. The cloacal membrane separates the cloaca from the proctodeum of the anal pit. During development, a sheet of mesenchyme urorectal septum developed to divide the cloaca into a ventral, urogenital sinus, and a dorsal portion or anorectal canal. By week 7, the urorectal septum reaches the cloacal membrane dividing into ventral or the urogenital membrane and dorsal or the anal membrane portions. The anal canal, the epithelium of the superior two-thirds of anal canal is derived from the endodermal hindgut. The inferior one-third develops from the ectodermal proctodeum. The junction of these two epithelia is indicated by the pectinate line, which also indicates the approximate former site of anal membrane that normally ruptures during the 8th week of the development. The hindgut In the hindgut formation, the distal one-third of the transverse colon is descending colon, sigmoid colon developed from the cranial end of the hindgut. The upper anal canal also developed from the terminal end of the hindgut, with the urorectal septum dividing the upper anal canal and the urogenital sinus during the 6th week. By the 7th week, the urorectal septum fuses with the cloacal membrane, giving rise to the anal membrane. And the urogenital membrane, the anal membrane ruptures during the 8th week and allowing communication between the anal canal and the amniotic fluid. The superior two-thirds of the anal canal originates from the hindgut, and the inferior one-third is derived from proctodeum. The pectinate line is junction of proctodeum, ectoderm, and hindgut endoderm. So thank you for listening. That's all.
Good morning, Mom. Good morning, classmates. So, our topic for this video is all about the development of the digestive system from the primitive mesenteron, specifically in the digestive tract. Why is digestion important? Digestion is important for breaking down food into nutrients, which the body uses for energy, growth, and cell repair. Food and drink must be changed into smaller molecules of nutrients before the blood absorbs them and carries them to cells throughout the body. Digestive glands include the following: esophagus, stomach, duodenum, pancreas, liver and biliary apparatus, spleen, midgut, hindgut, anal canal. Esophagus. The trachea the tracheoesophageal septum divides the foregut into the esophagus and trachea. Stomach. The primordium of the primitive stomach is visible about the end of the fourth week. It is initially oriented and suspended from the dorsal wall of the abdominal cavity by the dorsal mesentery or mesogastrium. During the stomach rotates 90 degree in a clockwise direction along its longitudinal axis, placing the left vagus nerve along its anterior side and the right vagus nerve along its posterior side. Rotation of the stomach creates the omental bursa or lesser peritoneal sac. Duodenum. The duodenum acquires its C-shaped loop as the stomach rotates because of its location at the junction of the foregut and the midgut. Branches of both the celiac trunk and the superior mesenteric artery supply the duodenum. Pancreas. The pancreas develops from two outgrowths of the endodermal epithelium, the dorsal pancreatic bud and the ventral pancreatic bud. During rotation of the gut, this primordial comes pancreas. The ventral pancreatic bud forms the uncinate process and part of the head, while the dorsal pancreatic bud forms the remainder of the head, body, and tail of the pancreas. The ducts of the pancreatic buds join together to form the main pancreatic duct, but the proximal part of the duct of the dorsal pancreatic bud may persist as an accessory pancreatic duct.
So my topic is liver and biliary apparatus. So the liver develops from endormal cells that form the hepatic diverticulum. So the liver grows in close association with the septum transversum, with later form part of the diaphragm. As it grows, the hepatic diverticulum divides into a renal part, which form the parenchyma of the liver, and the caudal part, which gives rise to the gallbladder and cystic duct. So the hematopoietic cells, Kupffer cells, and connective tissue of the liver are derived from mesenchyme in the septum transversum. The embryonic liver is large and fills much of abdominal cavity during the 7th through 9th weeks of development. So blood formation, hemophysis, begins in the liver during the 6th week of development, and bile formation begins in the 12th weeks. So the bile is the greenish brown thick substance produced in the liver and is stored in the gallbladder. So the bile function is to be released into the small intestine when the fatty meal is ingested to help break donor fats for absorption. So next is the spleen. The spleen develops from mesenchymal cells located between layers of the dorsal mesogastrum. So next is the midgut. The midgut communicates with the yolk. The yolk, as the midgut form, it elongates into U-shaped loop. Midgut loop, the temporary project into the umbilical cord, the physiological umbilical herniation. So the cranial limb of the midgut elongates rapidly during development and forms. So the jejunum and the cranial portion of ileum, the caudal limb forms and cecum, appendix caudal portion and the transverse colon the transverse colon. The caudal limb is easily recognized during development because of the presence of the cecal diverticulum. So the midcut loop rotates 270 counterclockwise around the superior mesenteric artery as it retracts into the abdominal cavity during the 10th week of development. So the midcut develops into the distal jejunum and jejunum, ileum, and cecum appendix, and ascending colon and proximal transverse colon. So next is the head cut. The head cut is defined to begin where the blood supply changes from the superior mesenteric artery to the inferior mesenteric artery at the distal third of the transverse colon. So this is the mid cut and this is the head cut. So partitioning of the cloaca. So the cloaca is the endodermally lined cavity at the end of the gut tube. It has a diverticulum into the body stole called allentous. So the cloacal a diverticulum into the body stole called So the cloacal membrane separates the cloaca from the protodium or anal pit. So during the development, a sheet of mesenchyme, so urethral septum, so developed to divide the cloaca into a ventral urogenital sinus, so and a dorsal portion of anorectal canal. So by week 7, the urethral septum reaches the cloacal membrane and dividing into ventral urogenital membrane and dorsal anal membrane portion. So next is the anal canal. So the epithelium of the superior two-thirds of the anal canal is derived from the epidermal head cut. So the inferior one or third of the anal canal is derived from the endodermal head cut, and inferior one or third develops from the ectodermal prodigium. So the junction of these two epithelia is to indicate by the pactinate line, which also indicates the approximate of former site of anal membrane that normally ruptures during the 8th week of development. So that's all my report. Thank you for listening.